So a very good afternoon, everyone, and welcome to the second vertical. It's a quick change from the first to second today. So in today's talk, we have with us Dr. Anoop Kumar. Dr. Anoop Kumar is currently working as an assistant professor in Department of Pharmacology and Toxicology at NIPER Raebareli. Earlier, he has worked as officiating head and associate professor in Department of Pharmacology, Indo-Soviet Friendship College of Pharmacy. Very good afternoon, everyone, and welcome to the second vertical. A quick change from the first. He has also worked as a research scientist in Medical Affairs and Clinical Research Department of Sun Pharmaceuticals Limited, Gurugram, and Translational Health Science Institute, Faridabad, India. He has completed his master's from Jamia Hamdard, Delhi, and PhD from Birla Institute of Technology, Mesra, as a DST inspired GRF and SRF fellow. Its major research interests are repurposition of drugs against various diseases using in silico, in vitro, and in vivo techniques, as well as risk and benefit analysis of medicines. I'd like to thank Dr. Anoop Kumar for coming here, and this is his third session. He has been delivering in the DDH training program. So with this, I'd like to hand over the session to Dr. Anoop Kumar.
Thank you, sir. Sorry. Thank you, sir, for such a nice introduction. So today I am basically going to talk on ADMET prediction using online web server. So in drug discovery hackathon, particularly in phase 1, we are looking for the screening of number of molecules which are available or molecules which are new one or molecule which are withdrawn from the market or some medicinal plant databases, and our major objective is to come up with a some promising molecules which we can test in experimental studies, or we can do animal testing. So the what should be the criteria for the molecule? Which one is best? Suppose you have done docking studies of various databases, the molecules which are available in the databases, and you come up with the three promising chemical entities. You are saying, sir, these are the most promising on the basis of their doc score. So the next question will be related to their ADME as well as the safety profile. So the molecules which are available in the market, and when we are going to submit the IND application to the regulatory agency to start a clinical trial, we have to show that the molecule is safe as well as effective in manners. So safety of the molecules, we have to do number of experimental studies, but before doing the experimental studies, we can predict the toxicity of these molecules. If suppose the molecule is showing very good binding affinity towards target, what we are predicting it's likely be a toxic into a number of targets. So we have to be think of how to reduce the toxicity of these molecules at early phases of drug discovery. So it can reduce the time as well as save a lot of money which is involved in the drug development process. Another thing is the molecule should be efficacious, and as I told in my previous lectures also, efficacy is indirectly related to pharmacokinetic profile. Pharmacokinetic means how our drug is whenever we are administering a drug to a body. So how our body is behaving against that molecules, that is we call kinetics. So what happened to the drug during the movement in the body, that is the pharmacokinetics. So the response of any drug is depend upon the how much concentration of the particular molecule reach at the target tissues. So that's why it's indirectly related to efficacy. If our molecule is not able to reach at the desired concentrations at the therapeutic targets, it will not going to show the efficacy. So the molecules which is efficacious, it should have also the good ADME profile as well as the toxicity profile. And these ADMET profile, that is absorption, distribution, metabolism, excretion, and toxicity, we can also predict by using commercial available software, as well as there are a few lots of online web server also by which we can predict these things. So in this talk I am going to talk about what is ADMET, what are the different online web servers available by which we can predict the ADMET profile of the molecules, and how to analyze the results, followed by the conclusion. So first, as I told you, ADMET, A is absorption, distribution, metabolism, excretion, and toxicity. So if you see this figure carefully, so particularly when see as a patient's the most preferable route is the oral route. Oral route. The reason is because patient compliance is much more. Whenever we are converting the molecules into a suitable formulation, first we are thinking whether that molecule is able to convert into a oral formulations or not due to the patient compliance. So in oral route, the molecule will has to cross various barriers. It has to reach the gastrointestinal tract. In stomach, pH is acidic, then followed by the basic pH in the intestine. So the molecule should be stable at the different pH, and the molecule should be able to cross the cell membrane barriers. GIT cell membrane barriers in intestines. There are microvilli which provide the large surface area, so that's why most of the drug are absorbed from intestine. The molecule should able to cross these membranes, and then through these through portal vein, the molecule will reach to the livers, where the metabolism of that molecule will happen. That we call first pass metabolism. And if you see, there is a gut wall. This is the efflux. There are a pump also, efflux pump. So efflux pump, particularly what these pump do, they efflux the intracellular drug outside the cells. So if our drug will uh, efflux by the efflux pump outside the cells, then it will reach back to the intestine, and ultimately it will excrete it out through the feces. So that molecule will not ultimately reach into the systemic circulation and not reach to the target site. So that molecule will not show the action. So we have to see whether that molecule is able to cross this barrier successfully, how much efflux can be happen. uh, so ultimately it will reach to the liver, where metabolism will happen. As many of you knows about, there is a phase 1 and phase 2 metabolism reactions, by which molecules are metabolized, and some molecules are more metabolized, so their concentration in the systematic circulation is decreased. Some molecules are less destroyed. So depending upon the molecules, we have to see which molecule is going to be metabolized by which enzymes and how much it will be destroyed. And ultimately that molecule will reach into the systemic circulations. And once molecule reach into the systemic circulations, it's not like that molecule will going to reach only on the target sites. You see, the through the systemic circulation, that drug molecule will reach to the all the organs, brain, liver, kidney. It will circulate all the systems, and depending upon your structure of the molecules or what you have designed, it will bind to the target receptors. So the molecules we are designing in such a way that will bind particular to the targets, that is we called on levels. But practically it's not like that. When we are giving a molecules, it's going to, some part of the molecules are also going to bind with other tissues, because through systemic circulations that molecule will reach to all the organs. So it will going to bind with others organs also, other cells also, depending upon its structures or nature of the molecules. If it's a more lipophilic, it can cross the blood brain barrier and reach to the brain. So and produce the undesirable effects, that is we call toxic effects. So these ADME absorptions, how the molecule will distributed, how it will be excreted, and how it can show the toxic response, all these things we can predict by using online web servers. There are a number of web servers by which we can predict. So the question is why there is a need to do the predictions of ADMET. Why there is a need these to predict the ADMET profiles, and when to predict means either we have to be predict the ADMET profile in phase 1 before phase 1 clinical trial, or in drug discovery phase, or in phase 2 trial or phase 3 trials, where we have to be predict, where we have to use these tools to predict the toxicity profiles of the molecules. So see, first thing is why I needed to do these studies. there are a couple of reasons, and this is, if you see these diagrams carefully, you will find that this is the diagram which is showing percentage of the molecules which are failed in preclinical trials and the molecules which are failed in the clinical trial, and these are the percentage which shows the reasons behind their failure. So if you see, this red line is showing pharmacokinetics, a molecule 5% is approximately 5% is failed due to lack of efficacy, some are due to animal toxicity, some are due to adverse effects in men. So these all contribute to stop that molecule to go further to further phases. So if you see, most of the molecules are failed due to their poor pharmacokinetic properties. However, this data is very old. In the, if you see, it's a 1988. So this percentage might be now changed due to early predictions of these activities through computational tools. So, but again, lot of molecules are failed in later stage of drug development due to their poor pharmacokinetic profiles. In clinical trial also, when we have studied everything in the animals, everything in the in vitro followed by in vivo system, and when we put the molecules in the phase 1 clinical trials, lot of molecules are failed due to their poor pharmacokinetic properties. A lot of molecules are not absorbed in the human. So ultimately it will lead to a loss of lot of monies and as well as the time which is involved in the process. So these computational studies could help to reduce these attrition rate, means it could be much more helpful. So that is the one reasons why there is a need to do these computational studies for the predictions. Another reason is these computational studies will decrease unnecessary testing of lot of molecules. Suppose you have designed number of analogs. So by computational studies, you can screen that analogs, which one is most promising, and you can synthesize only the analogs which are most promising. Means you have done the docking studies, you have done the toxicity studies, kinetic predictions, and on the basis of that one, you make a decision which molecules you have to be go further. So you can, and finally you are going to synthesize only that molecule. So it will reduce unnecessary testing of the molecules also, or synthesis also. Another question was when to predict. So the question is, at what phase we have to use these tools. So these tools we have to be used in the early drug discovery phases. The reason is, see, in to do a computational studies, there is a not need to do much more things. Means there is no need to do to collect much more things. Only what is required, only you need the structure of the molecules. There is no need of chemicals. There are no need of anything. You need structure of the molecules, and by the structure of molecule, and you can predict the uh, pharmacokinetics, safety profile, efficacy profile of that molecules. So on the basis of that results, you can decide whether you have to be which molecules you have to go further or not to go, or whether you have to be modify the structure of the molecules to make it more absorbables, or whatever the problems you will face. Accordingly, you can modify the structure of the molecules. So these are the couple of the reasons. Another is popularity. The question is how many studies are available and what is the trend of the studies in the literature regarding ADMET studies. So I have just collected the data from the PubMeds and just plotted it with bar diagrams in the Excels, and you can see from 2010, 11, 12, 13, there is a not much more increase in these type of studies, but after 2015, 16, 17, 18, 19, there is a much more increase. Various researchers are using this tool for the predictions of pharmacokinetic as well as toxicity profile of the molecules. In 2020, already a number of studies has been done, or it will be increased further also. So this is just a trend, popularity of these ADMET studies. So what parameters you have to do in ADMET studies? As I told you, first thing is absorption, whether your molecule is going to absorb or not. That is the first things. So when, particularly when we are giving the molecule through a oral administrations, so when molecule you will give, then that molecule should able to cross these membranes, and it depends upon number of factors. It depends upon molecular weight of that molecules. It depends upon nature of the molecules, whether your molecule is lipophilic or hydrophilic. It depends upon number of rotable bonds. There are a number of descriptors for on which this absorptions of the molecules is depend, and after that it reached to the blood vessels or systemic circulations, then it will distribute it for various, it goes to the metabolism. It can produce the desired action. Suppose you have designed a molecule for the nervous system, then it has to be crossed the blood brain barrier and bind there and shows the actions, and then finally it will excrete it out. So this first thing is absorption. So when you will use various tools, you will maybe many of you have heard about Lipinski rule of 5. So there are various factors which determining drug transfer across the cells. First, the molecular size of drugs. As you know, most of the drugs which are available in the market are around 200 to 500 Daltons. Solubility, see, if you are making a very much hydrophilic drugs, it will also not cross the cell membranes. It will not reach to the target site. If you are making more lipophilic, it will also not cross, or it will accumulate in the bodies and produce the toxic effect. So there is a balance is required. Route of administrations, if you are giving the drug through IV route, so then absorption phase is not there. So you can, the IV route means you are directly injecting that molecules into the blood. So only distributions, metabolisms, excretions, and toxicity you can predict. Influence of pH. pH also plays a significant role in the absorptions of the molecule. As I hope many of you knows, the weakly acidic drugs in acidic media absorb faster due to their predominant unionized form, whereas weak basic drugs are absorbed from the intestine. So it depends upon the molecules. So as I told, first descriptor is molecular weight. So most of the drugs, as I told, 250 to 450 Dalton or 500 Daltons, but there are some light ones are also like nitrous oxide 44, ethanol 46. There are drugs which are quite heavy like tuberculin. There are some drugs like peptides that are much more larger, so like antidiuretic hormones are still larger, 1000 to 2000. Insulin is a well known is small protein 6000. So if you see the size of albumin, that is the plasma proteins, it's a more than 60, around 65,000, and other proteins are still bigger. So is it means if molecular size is more than 500, is that molecules will not cross the cell membrane? They will cross, but they will cross through a different mechanisms. The molecules whose size is between 200 to less than 500 Daltons, they are easily cross the cell membrane through passive diffusion. There is no need of active transport. So only due to concentration gradient difference that molecules will cross the cell membranes. So molecular weight is much more important when we are designing a molecules. So we have to be look for its molecular weight. If molecular weight is much bigger, then we have to be think of by which process this molecule will cross the cell membranes of the GIT tract or the systems where it's going to be act, right? Another thing is lipophilicities. Lipophilicities also plays an important role. As I told, if we are making a drug much more lipophilic, it also not cross the cell membranes. So before lipophilicities, I have just added one slide about the cell membrane or plasma membrane. So you can see these consists a lipid bilayers. There is a hydrophilic ends. Heads are oriented outwards. Hydrocarbons tail are oriented inwards to form a continuous lipophilic hydrophobic phase. So these makes a cell membrane. So molecules should have the ability to cross these hydrophilic parts, and once it cross the hydrophilic part, it reach to the lipophilic part. So it should able to cross this lipophilic part, and then again it will reach inside. So you can imagine this should be in the balance. Means it's not like that you are making much more hydrophilic. If you are making a drugs, it will is excreted very quickly from your systems. So if you are making much more lipophilic, they can retain in the body for a longer period of time and can produce toxic effects. So we have to make a balance. Apart from that, in the cell membranes, there are a different channels also for the ions or water soluble molecules. There are some transporter also. This is the carrier transport system by which molecules are transferred across the cell membranes. Apart from that, on the cell membrane, there are receptors also, like your GPCR, that are cell membrane receptors. Ligand gated ion channels are also on the surface. So these all things are on your cell membranes, right? So number of molecules. So another thing is number of molecule crossing membrane per unit area per unit time. It depends upon permeability coefficients, and permeability coefficients we can measure by calculating the POYW. I hope many of you have heard about POYW coefficients, means oil by water coefficients. So solubility in the lipid membrane, partitions coefficient between membrane phase and the aqueous environment. So you can predict this log p values also by using these web server. Diffusivity, a measure of mobility of molecule within the lipid expressed as a diffusion coefficient. Another things on which the number of molecule crossing per unit area per unit time is concentration gradient across the cell membranes. See, in the passive diffusion, the molecules are moved across the cell membranes from higher concentration to lower concentration. That is we call passive diffusion. A drug with very high, as I told, very high lipid water partition coefficients will generate a higher gradient which favors more rapid diffusion across the membrane and into the cells. But when you are making a very much lipophilic, then it will retain in the cells. It will not easily excreted out. Diffusion coefficient is inversely related to the square root of molecular weight. So you have to be consider all these factors when you are predicting ADME profile, particularly the absorption profile. So this from this diagrams it will be more clear. If we are making a lipophilic molecule, see this is just a scales. This side there is a hydrophilicity, blue side is hydrophilicity. So you have to be make your molecules in this range, means neither more hydrophilic neither more lipophilic. So if you are making a more lipophilic, it will be absorb much more access to the brain. It depends upon if you are going to develop a molecule for nervous systems, then it should have the ability to cross the triple V. So depending upon the nature of the disease which you are targeting, not released from albumin, rapidly metabolized, less excreted by kidney, hydrophilic, not well absorbed, well distributed, metabolically stable, but rapidly excreted. So optimum of lipophilicity for an oral drug. So that we call drug likeness. So drug likeness, whether suppose you have 100 molecules designed, you have designed analogs of 100 molecules you have, and now you would like to know out of 100 how many have drug likeness properties or how many have not drug likeness. So by simple using these servers, you can predict whether these how many are have the drug likeness property, how many have don't have. uh, Only basic concept is on the basis of your Lipinski rule of 5. There are a few examples by which molecules which are earlier discovered and after that it has been observed that that molecules is not absorbed properly. So scientists have make some modifications in the structures and then discovery of new molecules. So well-known example is tioconazoles. This is a poor effect orally, so scientists have modified these molecules to make it more absorb orally, and that molecules is known as fluconazole. HIV protease inhibitors, Merck has developed these HIV protease inhibitors initially, but that are poorly soluble and not able to cross the cell membranes. So they have done the modification in that moieties of these molecules, and they have incorporated basic amines, and that drug is known as indinavir for HIV protease inhibitors. So these are a few examples by which initially you have see if you have predicted some molecules and you are saying, sir, this molecule is showing optimum binding affinity towards the target, but problem with this molecule is it's when we are predicting the toxicities or predicting the ADME profile, it's not qualifying. So that doesn't means you have to be stop working on that molecule. You have to be think how you can minimize uh, these toxicity problems, how you can modify the ADME parameter as per the requirements. So accordingly you can modify the structure of the molecule and make it more absorbable and less toxic and more efficacious. So these drug likeness that I told, drug likeness, so these all properties we can predict by using freely web servers also, some are available commercially also. So these all things we can predict: drug likeness, ADME, toxicity predictions, and these all predictions are depends upon the calculations of molecular descriptor. See, every molecule have a different descriptors. If I give a simple examples, molecular weight is also the one of the descriptor of the molecules. Log p value is also the one of the descriptor of the molecule. So on that basis, you can make a prediction. And well known is your QSAR studies. If you go through a DDH training program, you will find lot of lectures in detail about the QSAR studies, where you will get to know how to calculate these molecular descriptors on the basis of this one, how to create a model and then validate for the predictions. So drug likeness prediction, first when we are predicting ADME profile, first when you put your submit your molecule for the predictions, you will get various values like moleculars, weight, log p values. It will tell you about the how many number of hydrogen bond donor in that molecules, how many number of hydrogen bond acceptors. So these all tells us about the Lipinski rule of the 5. And according to Lipinski rule of 5, if the molecules are less than 500 daltons, the log p value is less than 5, or less than 5 hydrogen bond donor, less than 10 hydrogen bond donor, so that molecules have more chances to absorb orally when you will test experimentally. But more chances, it's not 100%, means chances are much more when the molecules are following Lipinski rule of 5, that will be absorbed orally. But sometimes if you see many drugs are in the market which are not following the Lipinski rule of 5, but they are available in the market as a oral formulation. So there are some sorry. So there is some exceptions where Lipinski rule of 5 not work. So exceptions I am going to discuss. This is just a one thing is in Lipinski rule of 5 is dependent upon the it will predict only when your molecule is going to cross the cell membranes through a passive diffusion. If your molecules are going to transport across the cell membrane through active process or any other process, so this Lipinski rule of 5 will not work. So this diffusion, passive diffusion, I hope every one of you knows, this is the flow of the molecules from higher concentration to lower concentration. That is we call concentration gradient. From higher concentration, molecular will move, and movement stop when the number of molecules on both sides will reach the equilibrium. So automatically that movement will stop. So if your molecule is going to pass the cell membrane through passive diffusions, then Lipinski rule of 5 will work. So drug likeness prediction. So there is some modification in Lipinski rule of 5. As I told, there are four parameters: molecular weight, number of hydrogen bond donor, number of hydrogen bond acceptors. Or apart from that, later on, some scientists have done little with additions of other parameters, like they have added polar surface area, that is called PSA, total polar surface area, molar refractivity. So all these features of the molecules. So these are Weber, Egan. So by using these online predictions web server, you will get to know about Lipinski, as well as if you are getting the value, not value, if you are getting the information about Gauss, Weber, Egan, so don't worry, that is also tells us about the drug likeness predictions, means the molecules is have the drug likeness characteristics or not, right? So, as I told, rule of 5 or this Lipinski rule violations are antibiotics. If you submit the structures of the antibiotics for the prediction of Lipinski rule of 5, you will find that uh, it not follow number of these the molecular weight is big, your number of hydrogen acceptor is more, number of hydrogen donors are more, so it's not following the Lipinski rule of 5. Antifungal molecules, vitamins, cardiac glycosides, the molecules which you are giving through a IV route, the Lipinski rule of 5 will not work. The reason is because there is no absorption phase. If you are giving a molecule directly to the systemic circulation through injection, so obvious the absorption phase is not there. So Lipinski rule of 5 will not work. Lipinski rule of 5 is just telling you whether your molecule is going to absorb or not. Natural molecules are also not following the Lipinski rule of 5. So these there is a possess if the molecule possess a group, but it doesn't means if the some molecule not following the Lipinski rule of 5, it doesn't means that molecule will not absorb orally. There is a more chances it will not absorb, but you can't say there is a 100% it will not absorb. I told you these examples also where Lipinski rule of 5 is not followed, but they are available in the market as a oral formulations also. So these possess group which act as a substrate. Suppose some your molecule has a some group which act as a substrate for the transporter. So in that case also your Lipinski rule of 5 will not work. It will work when your molecules are crossing the cell membrane through passive diffusion, as I told earlier. So this is the first steps, drug likeness characteristics. So in drug likeness characteristics, when you submit to a online web server, you will get all these values: molecular weight, number of hydrogen bond donor, number of hydrogen bond acceptors, molar refractivity, or your polar surface area. So all these values you will get, and these values will tell you drug likeness characteristics, right? Now if you would like to know whether your molecule will permeable or not in intestinal cell lines, or particularly in experimentally we are checking the molecule is going to pass through a colon cancer cell line or not, or MDCK cell lines or not. So by using computational technique also we can predict the same, whether our molecule has ability to cross these CACO cell lines, well-known cell lines for the permeability studies. That is the colon cancer cell line. This is the kidney cancer cell line, other one MDCK. It will also tell us about whether our molecule has ability to cross permeability or not. Human intestinal absorptions, it can also be predicted. Skin permeability levels we can also predict. Another things we can predict about the P glycoprotein substrate or inhibitor. As I told in my first slides, there is some efflux pumps are there. So these efflux pump, what they do, they efflux the drug outside the cells. So ultimately your target concentration of the drug at the target sites will be reduced. So you can predict whether your molecules is going to inhibit that P glycoprotein inhibitors, or it will affect by these P glycoproteins. So molecule travel across as in absorptions. As I told, molecules who are going to cross the cell membrane through passive diffusions, that we have to consider in the Lipinski rule of 5. So the another questions might be arise in your minds. So how we will came to know that which molecules will cross through a passive diffusion, which molecules will cross through a active diffusion, which molecule will cross through a facilitated pinocytosis process or phagocytosis process. So by just calculating the physiochemical characteristics of the molecules, you will get a sense this molecule is going to cross cell membrane through which process. So if your molecule, this is a cell membrane, as I told, there is a hydrophilic ends and these are the hydrophobic ends. So suppose your molecule is small, means molecular weight is less than 500 dalton, and it's a uncharged, means not charged. So that type of molecules will easily cross the cell membrane through passive diffusions, right? Like your molecules are water, urea, carbon dioxide, oxygen, nitrogen. So they will easily cross. But if your molecule is large, but okay, it's uncharged, so like your glucose, sucrose, so it will not cross the cell membrane through passive diffusion. But it doesn't mean these molecule will not cross the cell membrane. They will cross the cell membrane, but through other process like active process. They need the energy to transport. So if your molecule is small, another scenario, if your molecule is small but it's a charged ions, so it will also not cross the cell membrane through passive diffusion, like your hydrogen ion, sodium ion, potassium ion, calcium. They will transport through carrier mediated or active transport systems. So depending what I want to say, depending upon the nature of the molecules, we will get a sense through which process it's going to cross the cell membrane. And according to the process, you can go for the Lipinski rule of 5 predictions, whether it will help or not, depending upon your molecules physicochemical characteristics. So another thing is, after absorptions, as I told, the drug will reach into a systemic circulations, and once it reaches into a systemic circulations, it is going to distribute to different tissues, different organs. So we can predict about the distributions also through using online web servers or through commercial available softwares. If you would like to know whether your molecule is going to cross blood-brain barrier or not, you can predict log VB values, and from log VB values from these predictions, you can get to know whether your molecule is going to penetrate blood-brain barrier or not. So these are some values can readily penetrate the blood-brain barrier if this value is diffused into the brain. So depending upon your targets, what you are going to target, whether you want to reach the molecule into the brain, sometimes we don't want to reach the molecule into the brain to reduce the adverse effects, to reduce the side effects in the brain. Sometimes we want is such a type of molecules. If we are working on a brain, we are working on brain disease like Parkinson, Alzheimer, or such type of disease. So we want the molecules should have the ability to cross the blood brain barrier. So depending upon the targets, we can predict whether our molecule is going to cross or not. And after whatever the objectives of the studies, accordingly we can modify, and then after the modification we can again do the predictions of that modified molecules, and we can see whether after doing the modification in the molecules, whether its log BB value is improved or not. Whether the earlier suppose you have done the modifications, you have increased the log BB value, but your molecules now become mutagenic. You have predicted it's a mutagenic, earlier it's not a mutagenic by doing that substitution. So you have to be carefully analyze the data after modifications, what effect on the other parameters also. Volume of distribution sorry, volume of distribution, as I told in my previous lectures also, volume of distribution tell us about where in the bodies the drug is located. So accordingly, according to volume of distribution, it's suppose it's around 4 liters, so you will get to know the drug is in only in the plasma. If you are getting much more value of volume of distributions around 40 liter, 42 liter, so you can understand the molecule is inside the cells, intracellulars. So by getting to know about the volume of distribution, or by predicting the volume of distributions, we will get to know where in the body drug is located. So another is after distributions, because after distribution drug will bind with its desired targets, or also some of undesired targets, and shows its sections, and finally it will excreted, metabolized, and metabolisms. You know, C by P, cytochrome P450 is the major enzymes, and there are various isoforms, C by P2B6, C by P3A4. And these metabolisms we can predict by using these servers. We can predict whether the molecules which we have designed is enzyme inducers and which enzymes it's going to induce, or it's enzyme inhibitors. See, if you can see the enzyme inducers drugs are like your grisofulvin, phenytoin, carbamazepine. These are the enzyme inducers. So if our molecules is enzyme inducer, then what will happen? You have to be think of uh, to increase the dose of your molecules, because enzyme inducers are the molecule which increase the metabolisms of other drugs. So you can understand, okay, with enzyme inducer, if our molecules will given, or our if our molecule is enzyme inducer, so what dose we have to be give. Same as for if you are predicting your molecule is enzyme inhibitors of that particular metabolic enzymes, you can predict whether your molecule is going to inhibit this isoform or this isoform or which isoform. And when you are giving clinically that molecules along with your molecules, so what can be happens? So it can inhibit the metabolisms of that molecules or your molecules. Then automatically you can think of in that case which dose you have to give. That will be called drug drug interactions. You can think of when finally you put your molecule in the market, so what drug drug interactions can be happens, so that we can predict through which isoform these molecules can be metabolized, or its enzyme inducer or inhibitor. So these things we can predict. Another excretions we can predict about the total clearance models. Clearance, as you know, clearance is the your drug removals per minute. So we can predict what is the clearance level of that molecules. We can predict the renal OC2 substrate. This is the renal uptake transporters, which is present on PCT, proximal convoluted tubule. This OC2 is a organic cation transporter. So we can predict whether your molecule is going to excrete much more faster or slower or whatever. So this is the ADME, means absorptions. As I first, drug likeness, we can predict whether our molecule have drug likeness characteristics or not. Then we can predict the absorptions, intestinal absorption, Kako cell lines, or these MDCK cell lines permeability predictions. Then we can calculate the after absorption distributions by predicting the VD values. Then metabolism we can predict through which metabolic enzymes our molecule is going to metabolize, and excretion we can predict. And now next is your toxicity, whether your molecule, the molecule which we have designed is toxic or not. These are some of toxicity I have mentioned, but many toxicities you can predict. There are number of servers by which we can predict all types of toxicity. We can predict even LD50 values of the compounds. We can predict all these. Hurj is the one of the important predictions in the toxicities. As you know, many drugs has been withdrawn from the market due to their cardiotoxicity. There are a number of examples, the drugs which are withdrawn due to their cardiotoxicities, QT prolongations. These are the problems which results in the withdrawal of the molecules from the market. So now what the most of the pharmaceutical companies are doing, they are predicting the cardiotoxic potentials of the molecules in the early drug discovery phases. They are going to predict, and this is the Hurj predictions, followed by in vitro assays. You can do this Hurj assay under in vitro conditions, so you can verify whether the prediction is wrong or right. So, but initial drug discovery phases, you can predict whether your molecule is going to effect on cardiovascular systems on hearts, whether there is any adverse effect of that molecule will be there or not. So this Hurj is a human ether-gogo related genes, and it encodes your potassium channels. So ultimately, if your molecule is going to inhibit this, interfere with this Hurj, so there is a more chances of QT prolongations. So that we call Torsady points. So these means your molecule have the potential to produce the cardiotoxicity. So you can take initial step to reduce these cardiotoxicities. Like another, you can predict the mutagenicity, hepatotoxicity, skin sanitization, carcinogenicity. We can predict nephrotoxicities. We can predict. So all these major toxicities we can predict, rather than we can predict others toxicities, also neurotoxicities. All these toxicity we can predict. So now this is the last part, the basic things about what are the online web servers and which servers are available for these predictions. So I hope you understand what is the basis of predictions, why we are doing these prediction ADMET studies, and these studies plays a significant role. Even though if you are saying your molecule is very good, efficacious, but your molecule is not having a good ADME or toxicity, less toxicity, so your molecules there is less chances to reach into a clinical phase. So online web servers which are available for these predictions, well-known servers, you can get number of list of number of web servers. Swiss ADME Predictor, Mole Inspiration, Pre-ADMET, PKCSM, Protox, Lajar, Osiris. There are many online web servers. So these servers predict these, whatever I have told you, the parameters. So these servers predict about the absorptions on the basis of this Kako cell line permeability, MDCK. They predict the drug likeness on the basis of Lipinski rule foil. So all these predictions this server do, and they are very easy to use. There is no need to install these installation of the softwares or these things. So you have to be just some online web server might be ask you about institutional login IDs, and then they will you can access this server. So these web servers are very easy to use. So all these web servers, basic principle of all these web server is, see you what you want to do. You want to do whatever the molecules you have designed. Now you want to check whether your molecules have good safety profile or good ADME profile or not. So or you want to predict the activity of the molecules. You want to predict whether your molecule is going to bind with suppose if I give you example of COVID-19 to RDRPs, protease, or nucleic acid protein or spike protein of COVID-19. So means we submit the molecules with unknown activity. We don't know about its efficacy and safety. So what these servers do? These servers depends upon the similarities. These servers has a library of the molecule with known activities. So when we submit the molecule in the servers, they look for the possible activities which will match with that structures. So it's very much important whenever you are using any web servers, read about it. What is its limitation? Because every web server, some web servers are designed according to only the clinical molecule, on the basis of clinical molecule. Some are only on the basis of preclinical studies. So depending upon their data sets, their predictions will be depends. So you have to understand. That's why there are a number of web servers are available. There are these are the few, but there are a lots of web servers you will find when you will do the Google about these online web servers. But basic principle is this, and according to their data sets, more the data sets you have, so more accuracy in the predictions of the activities. So that's why QSAR is much more popular. The reason is in QSAR we are comparing the experimental data sets with the predictive data sets. So means we are correlating with the experimental data sets. But in these web servers also we can predict this is the Swiss ADME predictor. If you just do a Google and type Swiss ADME predictor, you will get this Swiss ADME predictors, and just read about this web server. What is its means on what basis this web server is designed. You will get all the informations on any web servers. So in Swiss web servers, and as I told you, these web servers are very easy to use. You have to what you have to do. You have to just draw a structure of the molecules and just submit that molecule and run. In some web server you have to submit, and then you will get a results. But basic things you have to learn about whatever the web server you are using. on what basis it's doing the predictions, on the basis of clinical data sets or preclinical, or withdrawn molecules are included or not, what on what basis. So in Swiss ADME servers, you have to be just these are the drawing tools by which help you to draw a structure of the molecules. Yeah, and then you have to click here and then run, and after run you will get such type of results. So if you see carefully these results, you will find first thing is physiochemical properties. It's telling about the formula, molecular weight. So this is a important information. As I told you, if you would like to know this molecule have drug likeness characteristics or not, so you have to be see the what is the molecular weight. Then you have to be see the number of hydrogen bond acceptor, number of hydrogen bond donor, molar refractivity. As I told in after Lipinski rule of 5, there is a modification has been happened and scientists have been including molar refractivity. This is the total polar surface areas that has been included. So all these physiochemical properties you will get to know drug likeness characteristics of the molecules. This second, if you see, this tells about the lipophilicity or lipophilic characters of the molecules. So you can get log POYW, this is the octanol or water coefficients. So you can M log P values. So all these values will tell you about whether your molecules have lipophilic and how much lipophilic it is. So water solubility, as I told you, there should be a balance between the lipophilic and hydrophilicities. So you can get to know about hydrophilic as well as lipophilic characters. Pharmacokinetics, you can get to know whether your molecule is going GI absorption is low or medium. Blood-brain permeant, whether your molecule is able to cross the blood-brain barrier or not. P glycoprotein substrates, this showing no. These are the various isoforms C by P, 1A3, 2C19. No, that are not going to affect these. So all these drug likeness, Lipinski, Ghosh, Bever, Egan. So whatever the information you will get, you can make a conclusion whether your molecules need some modifications or not. So like that way, there are another web server mole inspirations. So you have to just do a Google, type in a Google mole inspirations, and you will get this informations. What is this mole inspirations, on what basis this web server is designed, and what is its limitations, and what you can do by using this web server. And these all are freely available. There is no need of any kind of license or any other thing. So if you see in these sites, you will get this Galaxy 3D generator, calculation of molecular properties, prediction of bioactivities. So you can predict, again you have to withdraw the structure of the molecule. As I told in my earlier slides, in computational studies, you only need the structures of the molecules or targets. What is your targets? And remaining things your databases will do, whether it's a soluble, not soluble, whether it is going to absorb or not. So you have to just calculate the properties. You can predict bioactivities also. Little bit you will get an idea. This mole inspirations, you will get mi log p. When you will click on mi log p, you will get more information what it is. You will get more information what is the range of TPSA, total polar surface area, what is the optimum values, and what you are getting. So accordingly you can see whether your molecules have good absorptions profiles or ADME profile or not. So there is another web servers pre-ADMET, which also predict. Just type again. You have to just type in Google pre-ADMET. You will get this pre-ADMET, and if you see this closely, there is a drug likeness prediction. As I told, drug likeness predictions on the basis of this Lipinski rule, lead-like rule, drug DB rule, ADME predictions. See, if I don't know it's visible or not. CACO2, MDCK, blood-brain barrier, HIA, human intestinal absorption, plasma protein binding, serum permeability. So all these things you can predict whether it's going to bind with the albumins or alpha-1 acid glycoproteins, whether it's permeable. Toxicity predictions, if you see this, there is a AIMS test that is for the mutagenicities, whether your molecule have mutagenic characteristics or not. If your molecule is mutagenic, then you have to be much more careful at early phases. and you can think of how to reduce the mutagenicity of these molecules. Then carcinogenicities also you can predict. So all these predictions you can do by these. Another is PKCSM is another web servers, where again you have to submit the structures. If you see this absorption, distribution, metabolism, excretion, toxicities, all these informations you can put molecule in the smiley formats. Also you can choose a file and upload it and then submit it to the server, and then you will get a results about these profile. As I told, Hers prediction models, many drugs has been withdrawn from the market due to their cardiotoxicities, due to their cardio toxic potentials. So, in early phases of drug discovery, we can predict whether this molecule is going to be a cardiotoxic or any adverse effects on the heart or not. So you have to be there are a Arch 1.0 server where you have to submit your molecule, and it will predict whether your molecules have this cardiotoxicity potential or not. So like that way, I have just included few web servers. All these web servers are very easy to use. That's why I have not included all. Otherwise it will become boring. So I have just your message is just there is very easy to use. You have to just draw a structure and submit. But when you are doing this, whenever you will get the data, just understands about the web servers, what is its limitations, and on what basis that web server is designed. And suppose you are going to predict the toxicities in rat or mice or in human, so accordingly you have to think of what you want, what is your objective, and accordingly you can choose which web server is the best for you. And another things what you can do, you can submit same molecule to 2 3 web server and just compare the results. Then you will get to know how close these results are and how much reliable these servers are. So just submit same molecule, do the same analysis by using 3 4 web servers, and just see if one server is saying it's a drug, this molecule have a drug likeness characteristics, another web server saying no, so that means there is a conflict. Then you have to see manually, you have to see the structure of molecules, means which one is correct. Means you can calculate number of hydrogen bond donor, hydrogen bond acceptors, you can calculate the molecular weight. So all the descriptors you can calculate. Also you can calculate in chemdraw also some of the descriptors. So accordingly you can do the comparison, and after the comparisons you can see the reliability and use these web servers. These are very helpful in early predictions of ADME as well as toxicity profile of the molecules. And after that, according to the analysis of the data, you have to be think of whether there is a need of any modifications where you can do the modifications, and that modification should not hamper the your efficacy profile. It should not uh, hamper the other other toxicities. Suppose you have you did the modification to reduce the carcinogenicities, but after modifications that molecule is become hepatotoxic. So you have to think of. So LD, there is a servers also for LD50 predictions. As you know, when we are dealing with the new chemical entities, or whenever we are doing some modification in the existing molecules, or we are going to repropose withdrawn molecules, so we have to calculate the LD50 values in rats or animals, because in human we can't do LD50. This is the lethal dose, means at which 50% of the population die. So it's not possible to do in a humans, but in animal studies we are doing to just to calculate the LD50. But now regulatory authorities are asking regularly to reduce the use of the animals. So in that case, these web servers will help to predict these values also, these LD50 values of the molecules. And well-known web server is Protox. Just read about it's also freely available through Protox. You will get to know LD50 values of the molecules. Again, you have to draw structures of the molecules and submit into the databases. and then see LD50, your the molecules LD50 is falling in what range, means less than 5, then it will be becomes class 1, class 2, class 3. So you will get a sense about the LD50 values of, and you can much more careful about the safety of the molecules. In protox, you can also predict about the targets, acute toxicity, organ toxicity. So number of things you can predict, but again you have to be see the data more closely, means more carefully, not closely, carefully. Uh, Means by using these servers you will get number of data, so it's very difficult which one you have to be keep. So you have to understand what that particular descriptor is telling us, so accordingly you can make a conclusions. So ADMET predictions, in particularly in early drug discovery phases, reduce the unnecessary testing. Earlier, what we are doing, we are synthesizing the molecule, number of molecules, medicinal chemists are synthesizing, and then after that they are going for a toxicity studies or efficacy studies, and came to know that that molecule is not working, or that molecule is toxic or less efficacious. So it means there is a lot of time consuming, also a lot of money is involved, resources are involved. So these web servers, or not web server, these computational techniques particularly helpful to reduce. We can design a different analogs, and we can predict the activities, as well as the toxicity, as well as the ADMET profiles of the molecule. So ADMET predictions in early drug discovery reduce attrition rate of molecule in early stage of drug development. Maybe one question might be arise in your mind, if there is a online web servers are available, so why we will use the commercial, why companies, particularly pharmaceutical companies, are using commercial softwares for do this these all predictions. So why there is a need to put such or purchase that type of software. So again, as I told you, the predictions depends upon how much data sets you have used for the development of that software. So the commercial software, obvious, they have put it large number of databases, so their reliability is much more as compared to online web servers. But at initial stages, we can predict, we can make a rough ideas about is this molecule is going to be absorbed better, or how it's going to be excrete, how it's going to metabolize, or is there is any toxicity problem. So finally, these studies help to reduce this development time, the discovery development time. But yes, you can't fully replace experimental studies. You have to do experimental study. But yes, if you have a 1 lakhs or much more designed analog, so practically it's not possible to do experimental studies for all. So these computational studies will help to predict at the early phases, so you can test minimum amount of the minimum number of the molecules in experiments. So thank you. Any questions? So
Thank you very much, sir, for this informative session on ADMET and web servers.
sorry, I wanted to acknowledge, I would like to thank Dr. SJS Flora, director of my institute, Naiper Raebareli, Vijay Raghavan sir, Shekhar sir, Anil sir, Abhay sir, Sastri sir, and all team member of Drug Discovery Hackathon. Thank you very much. Yes, sir.
Thank you, sir. Sir, we have received few questions, so with your permission, can we go ahead?
Yes, sir. Yes.
Yes, sir. Thank you, sir. So the first question is asked by Mr. Tanuj Saxena, and his question is there a difference in how drugs cross the blood-brain barrier with IP versus IV injections?
Yes. Yes, route of administrations always affect the absorption. As I told you, in if we are putting a molecule directly into the systemic circulations, there is no absorption phase. But yes, to blood-brain barrier crossability will depend upon the nature of the molecules. Whether your molecule has the capability to cross that barrier or not. So it depends upon your nature of the molecules, not on the route of administration. Suppose you have given IV as well as one injection IP. So in both route of administrations, your drug is reached into the systemic circulation, but after systemic circulations, it depends upon the molecules, whether that molecule have the ability to cross the blood-brain barrier or not. It depends upon that one. If that both molecules are reaching into the systemic circulations in same concentration through both routes. Generally, IP we are assuming 100% viability, and IP we will get less viabilities. But if it's a effective concentration, so again it depends upon nature of the molecules, whether that molecule is able to cross or not. It does not depend upon the route of administrations. Yes, sir.
Yes,
I
sir.
hope.
Thank you, sir.
Yes.
Yes, sir. Your explanation was very much apt to the point. And let me move to the second question. And it is also asked by Tanuj himself. And his question is it possible to lead a metallomolecule through the blood-brain barrier using carbon nanotubes without causing toxicity?
Sorry, sir, but he is asking.
Yeah, let me repeat it.
Yes, yes.
Is it possible to lead a metallomolecule through blood-brain barrier using carbon nanotubes without
Yes.
causing much toxicity?
Yes, it's possible to cross the molecule through carbon nanotube. Many research has been done in these areas, and many researchers have shown it will cross the blood-brain barrier. But again, it depends. Means we can't say about its toxicities, about its safety, because it depends upon which type of material has been used in that carbon nanotubes, and what type of molecule is crossing the blood-brain barriers. So if that molecule is going to do with any brain cells, it's going to damage the brain cells or not. It depends upon the nature of the molecules, which molecule is crossing through this carbon nanotube, and what type of materials the researcher has used to make that carbon nanotubes. Maybe that material, if it's not a biocompatibles, or means there is some problems with the materials, it can also lead to adverse effects in the brain. So we can't say we can cross, but we can't say it will be safe. It depends upon various factors, right? It does not depend upon one factors. You have to see number of reasons. Yes, sir.
Thank
Is
you,
it
sir.
make sense?
Yes, sir. That
Okay.
makes a sense of it. Yeah. So, sir, the next question is as there are a lot of clinical candidates which fail in the clinical studies or different clinical phases.
Yes.
So, is it mainly because of the toxicity issues due to which they are getting discarded from the main clinical phases? So, what is your opinion on, sir?
Yes. See, most of the molecules in clinical trials are failed due to their kinetic problems. First, many molecules we are getting very good absorptions in the experimental animals, but when we go for a clinical trial, same molecule will not absorb in the humans. And if the molecule is not absorbed or not reaching into a systemic circulation, so how it will shows the actions? So that's why there is a concept of phase zero clinical trials. So now what companies are doing, they are doing a phase zero clinical trial in which they just would like to know whether the molecule is going to absorb in the humans or not. They are giving the molecule in the subtherapeutic concentration. They are not giving on full effective concentrations or whatever they have designed, but they are using that molecule at subtherapeutic concentrations, and just would like to know whether that molecule is going to absorb in human or not, because number of molecule has been failed in phase 1 clinical trial due to their absorption problem. That is the one. Second thing is, if your molecule is absorbed in a phase 0 clinical trial, and when you have done the phase 1 clinical trials in a healthy volunteers, so it depends upon molecules. Many molecules also failed due to their safety problems. So, but majority of the molecules are failed due to kinetics, followed by their toxicity problems, and then efficacy problem is the last one. Because regulatory will ask you about toxicity, not about regulatory is not much about efficacy concerns. They will ask about the safety. What is the safety of these molecules? Whether it's going to affect this. So yes, as the question was, means only the toxicity is the reason to withdraw the molecule from the clinical trial. So it's not like that. Absorptions or kinetic parameters also plays a significant role, much more than toxicity, to withdrawn from the clinical trials. Right, sir?
Yes, sir. Thank you, sir. Sir, may I move to the last question for
Yes,
this session? So it is asked by Ms. Kavita Kumari, and her question is, do we have any ADMET filtered small molecule database?
ADMET filters.
Yeah, already the ADMET filters have been applied to those molecules. So is there any database which is existing?
Yes, the molecules, these FDA approved databases, these are the molecules which are already means past these all things, means drug likeness characteristics. That molecule have good absorption, that molecule have good kinetic profile. That's why they are approved and available in the market. So these approved molecules are available in databases, various databases, FDA databases. You will get the number of molecules which are approved and presents in the market. So that molecules are the standard one. Why that one you can compare your molecule is how much close it is. Yes, sir.
Yeah, thank you very much, sir, for all the answers being providing to us. And as we have reached the conclusion part of this session, I would like to thank you once again for joining with us in Vertical 2 session and giving this most informative session on ADMET and the open web servers for ADMET prediction. And so once again, on behalf of the DDH chairman, Dr. G.N. Shastri, as well as the entire team, we would like to give our heartfelt gratitude to you. And thank you very much, sir.
Thank you, sir. Thank you very much.
And thank you all the listeners on the YouTube as well as the MS team. And we'll be starting the Vertical 3 session today at 6 p.m.